Good morning, everybody. This is Sharan Ramaswamy, and welcome to today's seminar as part of Heart Month this February. It is my distinct pleasure and honor to introduce Professor Michael Sachs from the University of Texas at Austin. I had the pleasure of actually working directly with Dr. Sachs. Now it seems a while ago, I think over 10 years ago, while I was at the University of Pittsburgh, and so was he. Since then, Dr. Sachs has now moved to UT Austin. And Dr. Sachs is really a renowned world expert in heart valve mechanics, particularly in taking computational approaches to evaluating biomechanical properties and changes, specifically in a variety of valve-related diseases. And that is, of course, a topic of his talk today, which is entitled The Mitral Valve from Cellular Biophysics to Surgical Repair. Please welcome Dr. Michael Sachs and join me in welcoming him to today's seminar.
Great, thank you. Hope everybody's had a big cup of coffee because we have a long journey ahead of us in a way, because I've been studying heart valves since I started as my academic career almost 30 years ago. And a little interest before I get started, let me tell you, I went into heart valves because I did my doctoral work on the right ventricle. And at that time, there were many things we wanted to do on heart mechanics, but couldn't do. So I figured I'll work on heart valves to get myself started, and it'll be easy, and then I'll move back to the heart. And that was, like I said, about 30 years ago, and it's actually been a wonderful journey. They are extraordinarily complex structures. So let me go ahead and share my screen. Okay, and let me just start the slideshow here. Okay, Sharon, do you see the right one? You see the full screen?
Yes, we do. Thank you.
Good. Yeah, sometimes PowerPoint ends up giving you the monitor one. Okay, so this is really an effort to study the mitral valve in an area called the ischemic mitral regurgitation. But I think the one thing that, for those of you unfamiliar, most of with valve and valve structures and function, is that most of this work in cardiovascular is done with either the vascular system with blood vessels or in the heart, which are much further advanced. Our understanding of how valves work and how they function both biologically and mechanically is still really not well understood. And the field only really started maybe 20 years ago in terms of really trying to connect to the underlying biology of the valve and valve diseases. And much of this is because it's been traditionally dominated by surgical treatment, either by valve replacement or valve repair. And then as you dig into this, you'll find it's really pretty cool. So moving on. So first, I want to tell you a little bit about our center. It's sort of a joint operation between the Oden Institute for Computational Engineering and Department of Biomedical Engineering here at UT Austin. We have a number of collaborating centers and other universities, so we're a very collaborative group. The computational side is housed in the O'Donnell building, which as you see here, and then the wet lab we have is in the new engineering education and research building you see here on the left side here. That building, we just moved there about 3 years ago. Computational lab looks pretty much like any other computational lab, lots of desks, a lot of monitors, lots of great people. The wet lab is, as you can see here. And so part of what I've tried to do here at UT is to integrate these two very disparate approaches. And I hope today, for those of you who have an interest in both computational and experimental work, you can see it actually is possible to do, and in fact can be quite good. Our goal here at the center really is to develop ultimately tools for both a fundamental understanding, but computational modeling tools for clinicians to develop new therapy, and based on things that can now be measured, that even when measured even as little as 10 years ago. So it's a very exciting time to be in it. So we'll start with the valves of the heart. There are 4 total, as many of you know. There are the so-called semilunar valves, which are the aortic and pulmonary valve, which have a tri-leaflet structure. And then there's these really weird looking beasts called the atrioventricular valves, which is the mitral here on the left and the tricuspid on the right. And they these are their function is very basic. You know, it's essentially to direct unidirectional blood flow through the heart. But how they do it and the incredible mechanically demanding environment they're in is really um, quite astounding. That, for example, it's estimated that they will undergo 3 to 4 billion cycles um, in a single lifetime. And that level of, and for the most part, they work flawlessly, and that level of durability is not achievable by man-made structures, to my understanding. And this is because ultimately they're living structures. Uh, the mitral valve, which is our primary focus, is more complex than the atrioventricular valves. I mean, excuse me, the semilunar valves, because it's an integral part of the left ventricle. So you see there's this annulus where it attaches to the surrounding heart, but also attaches to the papillary muscles, which project out these cords, uh, which is actually involved not so much in helping the valve close, but it helps to reshape the ventricle during ventricular contraction. And here you can see a movie showing it working. Quite an amazing structure. So right away you can see this is a very complicated beast, and the interrelationship with the left ventricle is really paramount. Uh, the particular disease we're after is a very, very serious one, and it's unusual, unfortunately, very common. It happens when the left ventricle, of the patient will suffer from myocardial infarction, MI. And what happens after myocardial infarction is that the left ventricle will dilate, initially at first, and then much more later. Uh, and this dilation does two things. It causes the annulus to dilate, and it causes the papillary muscles to distend downward. And that sometimes will prevent the ventricle from, excuse me, the valve from coapting and forming a proper seal. So blood can leak back into the left atrium and back into the lungs, and ultimately cause congestion and heart failure. It can also cause stroke. It's a very, very bad disease. On top of the fact that the patient is already suffering from the effects of infarct. Now, the way this is traditionally done is what's called repair. Here's a 3D flow echo showing the regurgitant jet in the valve going back toward the left atrium. And after they do, they put a ring on it. It's actually open heart surgery. And it actually, you can see now that regurgitant jet has been stopped. So you would think, oh, that's great. Everything's all hunky dory. But the reality is it's not. And the great thing, there are actually two major ways to treat this ischemic mitral regurgitation is through repair, which tends to work very well initially, but has about a 50% recurrence of severe mitral regurgitation, sometimes as little as 3 to 6 months later. The other possibility is just to replace the valve with a prosthetic device. This can also get some good initial results, but there's continued problems with the fact that the valves don't last very long. And there's also a higher risk of death compared to repaired patients. So there really isn't a great way to treat this. The real issue is the repair. And part of the challenges clinically is that repairs tend to be done in a more one-size-fits-all fashion. And then what you really find out is that the disease is really quite specialized. So underneath all this, the question we were asking is, why do we get this recurrence? What is it? What's the mechanisms? And there's a number of things that go on in the valve. One of them is what goes on in the valve tissue itself. And this is all driven by the cellular population. These are called valve interstitial cells, which Dr. Hutchinson knows very well. And inside the valves, these are living structures. There are a endothelial layer, which we don't really still don't know too much of what it does. But the main one, the main cell types are the so-called interstitial cells, which are a complex, uh, basically a myofibroblast, and they're responsible for generally maintaining the valve extracellular matrix, and then also they become activated during disease processes and remodeling and so on. So ultimately, we think that much of what we find in the long term, again, we're not talking about short-term surgical outcomes, but what happens 6 months or greater down the road is driven by what happens to these cells. And there's a lot of evidence I'll be presenting today to show that. Now, when we look at the structure of the mitral valve itself, when you get into the leaflet, they're not just simple flaps. They're actually made of multi-layered structures. And this is some nice SCM work we did where we see this layer that faces the left ventricle called, of course, the ventricularis. And there's another largest layer called the fibrosa, which is made up of collagen. There's a less structured spongiosa layer, and then another layer that faces the atrium called the atrialis. Already you're seeing there's quite a bit going on here. And when we look, if this is a optical histology, kind of revealed the layers better. And you can see the fibrosa, if you look here, is it looks like a very dense weave of collagen. That's what it is. And its primary function is mechanical strength. And then if you look carefully in here, there are the interstitial. This is actually, you can see how layered and really complex it is. And then the atrialis, anyhow, there's a very, very different structure, very elastin rich. And the ventricularis has a structure similar to the atrialis, but inside that you can see this little guy here, and that's one of the interstitial cells, and they're completely embedded within this extracellular matrix. And the spongiosa now, of course, has an even less structured appearance. Now, this is a transmission electron microscope image of a mitral valve interstitial cell. You see the cell membrane, nucleus, sitting and completely embedded and surrounded by this extracellular matrix. And so the question is, ultimately, do they go, is both the ischemic mitral regurgitation and mitral valve repair, we think that in many ways what happens both in the ischemic mitral regurgitation, ischemic post-MI state, and then after surgery, the cells are activated. And you can see here that the collagen is here, the elastin is here, and the VIX are here, and all of this is highly coupled. And clearly, it suggests that what goes on at the organ level, that because of what happens with the disease, affects these cells and back. And so one of the first things we did is to say, okay, can we study this using a large animal model? And we did this with the Gorman Research Group at the University of Pennsylvania. They have a very well-established sheep model, male Dorset sheep, looked at about 8 valve, and they induce myocardial infarction by the ligation of the left circumflex coronary artery, and that creates about a 1/4 area of infarction. It's very reproducible model. It's been used for many years to study these things. And then we basically looked at what happens to the valve leaflet after 8 weeks post-infarction. Again, this kind of information is not possible in humans, and for time prevents, most of this has been published, and you're welcome to send me an email. I'll be happy to send you the papers. But we did a whole battery of studies to find out really just sort of what's happening just with the effect of ischemic mitral regurgitation alone. And we found all kinds of very interesting things. So this is a cross-section of this beautiful mitral valve in the normal state, and this is what happens in the 8-week state. And you can see already there's been some interesting changes. One thing is that the collagen goes up, GAGs and elastin go down. This is what happens if I go through that cross-section and look at a mass fraction. And you can see here that the collagen this is what the blue is what happens in the normal state. And you can see here what happens in the after 8 weeks. So basically what happens here is that these outer layers, the ventricularis, atrialis, spongiosa, really thin down. GAG content goes way down. Okay, so and then the elastin stays about the same, which you would expect because it's a very resilient protein. And so you can see already there's evidence of some profound remodeling, even just after 8 weeks. Look a little more carefully. This is some histological images. We notice that the control, this is the nuclear aspect ratio. So these little purple guys are the nuclei of those interstitial cells. And one way to estimate how much they're mechanically stimulated is look at their shape. And we do this by looking at the aspect ratio. In other words, the ratio of their long axis to their short axis. And you can see here, control what happens when you excise the valve. This is the pre-stress, I'll talk about in a little bit, and this is at the pre-stress. So what happens during the valve, as the valve's loaded alone, you can find that these aspect ratios will change quite a bit. And then if you look at what happens when we simulate it in vivo, that there's actually these big changes when you put, say, a ring on. So right away, we can see there's a very tight coupling between how the mitral valve physically deforms and how those cells are deformed. Going back to the histological results that we found in the 8-week period, we found on average that the cell nuclear aspect ratio actually went down, which suggests that the cells were actually less loaded with regurgitation. And in fact, there's a very nice correlation between how much the cells were deformed on average and the MR grade, that is how severe the regurgitation. Short of it is, the greater the degree of regurgitation, the more compressed the cells were. So already we can see a very nice correlation between these phenomena. Then we looked at mechanical studies. This is using some established techniques we developed many years ago. And we basically take sections of the mitral valve tissue, subject them to a range of physical examination. And what we find is that in the normal valve, if you look at the circumferential direction that's going around the valve and the radial, the valve, of course, is quite anisotropic, and that looks pretty normal. But just after 8 weeks post-MI, you can see here the radial and circumferential direction that you basically, the valve becomes nearly isotropic. So very profound tissue level remodeling. We then said, okay, we want to look into this more detailed. This was done in a live animal. Now we want to do is say, what really is going on in it? What is the link between this external tissue loading and what goes on inside the cell? So we conducted this study about 5 years ago, where we actually took sections of the anterior leaflet, mounted it on a little biaxial stretcher in a specially designed confocal microscope, so we could actually look at the cell population as we deformed the tissue. And you can see the setup here. And this is just what the collected data looks like. Again, there's the highly anisotropic behaviors. Now, what you find is actually really interesting. So what we've done here is that as we apply tension to that valve tissue, and this is an example of the cross-section we analyze, we actually can see how the cells deform as the tissue is loaded. And this is just an approximated in vivo load. And what we find is that the two major layers, the fibrosing ventricular layers, the cells deform very similarly, but in the other layers, they deform much less, although there is an overall, both exhibit the same overall trend of being loaded more, increasing the aspect ratio as you load it. But so what you're seeing is that the layer, that in with each layers, individual cells get loaded differently. So even within the valve, in the same layers, there is a major difference in how the cells in individual layers get stimulated. So it's really, that can explain some of the things we found in those in vivo studies. Now, we've been using computational methods, and I'll be getting to more of those a little later on in my talk. But one of the first things we're trying to determine is what happens to the valve when you just put this ring on, and is there anything that suggests an underlying biological mechanism? And the way we did this is that we did a study some years ago where we actually implanted sonocrystals on the surface of the leaflet and then looked at what happened when you put a ring on it. Very basic study. So, and then what we found was really quite interesting, which I'll show in a minute. But what we did then is to take those data and come up, that I showed you on the cell, and come up with this three-dimensional microenvironment model, so we can estimate what happens to the cells in vivo. Again, these measurements cannot be made directly, they have to be simulated. And so we created this microenvironment model to simulate the effects of ring annuloplasty. And that's what this is called, by the way. It's actually called undersized ring annuloplasty. I won't get into the details, it's rather involved. This was published in the Journal of Theoretical Biology about 5 years ago, but the short of it is we're able to use our very detailed knowledge of the extracellular matrix and mechanical behaviors and of the cells to determine how this all worked. And then we actually were able to simulate what happens in the loaded and unloaded state here, and you can actually see how much the tissue deforms, but also how much that deformation affects the cell population. And by the way, these golden guys here are the valve interstitial cells. The green is everything else, all the extracellular matrix, and that amount and orientation of the cells and their size is based on histomorphometric measurements. So these are actually a fairly accurate representation of what's actually going on. Now, how well does the model work? Well, we actually worked pretty well. So when we applied the same deformations we measured in vivo, the black line here is the simulation. The symbols are the data, and we can actually get a very nice agreement with the different cells in the different layers, even though they deform very differently. Interestingly, the basic modulus of the cell was actually really quite similar, and it's about around 5 kilopascals using this simple mechanical model. So what we're seeing here is that the cells all seem to have about the same physical properties, but their deformations are driven by changes in the extracellular matrix in the layer composition in the layer they're embedded in. So that actually was quite a nice insight, and this gives you a really kind of a nice example how you can use modeling to learn things that you can't get other ways. Now, going back to that ring study, what we found is that when you plant a ring, if this is simple, this is the radial deformation. The radial, by the way, is this direction here. Circumferential is this direction here. That the radial direction the ring doesn't do a lot, but in the circumferential direction, which is this direction, man, it really drops that strain way down. So cinching the valve or collapsing it down by putting the ring actually profoundly changes how that tissue is deformed over the cardiac cycle. So it's not just a simple close it down, get it to coap. It produces profound changes in how that leaflet deforms. And it goes way down by down by about 4 or 5%. Okay, so where are we now? So let me to give you an idea. So what we're seeing here is that mitral valve repair, ischemic mitral regurgitation are very complex phenomena, lots of things going on at lots of scales. And so the repair and the disease happen at the organ scale. Much of the pathology is driven by the cells. And our goal here is to try to link these two phenomena by moving down from the organ to the tissue to the cell, and the local extracellular matrix down to the cell, and then back up again. And then this is actually a very characteristic problem of most biological systems. There's lots of scales. Many of you may have heard the term multi-scale phenomena. This is a really great example of it. Our overarching goal, so the real question is, why are we doing all this? So the one is try to predict the recurrence of mitral regurgitation based on pre-surgical leaflet knowledge, deformation, and so on. And then the idea is that we can optimize surgical repair on a patient-specific basis and try to get away from the current surgical approach, which is one size fits all. So our hypothesis is this: abnormal organ and tissue level mechanical stimuli lead to non-physiologic interstitial cell deformations that result in phenotypic activation and altered biosynthetic activity. It's a very verbose way of saying it, but basically what it means is if you change if you change the leaflet patterns, you're going to change how the cells respond. That's basically it. Okay, so one of the first things we did is, okay, we're going to go down to the interstitial cell level and try to learn a little bit more about what these cells are. Now, one of the biggest problems we have is what is the exact microenvironment these cells are living in? And we got around, and this has actually not been studied, at least not directly, and we used a focused ion beam scanning electron microscopy technique, and we're able to, I think if this animation works, let's see, there we go. This is what it produces. So it's this whole serial sections of ion beam, and then we can reconstruct it. The red is the cell. Those are the elastin. That's the cell again. This is the nucleus here. This is a collagen fiber. Okay, and let me just go through this again. The blue there, we haven't separated, is the elastin. The gray is the collagen. So we're just melting that collagen away, and there you can see what it looks like in 3D. So already you can see that, well, this is really the environment the cell lives in. It's very complicated. And it's not simple one direction. This is actually a very complex three-dimensional problem. Here's just some other photographs of it. Here's the surrounding collagen fibers, and you can actually resolve the individual collagen fibrils. So this whole blue guy is one fiber, this darker blue is another fiber, and these little guys in it are the fibers. This is what the elastin looks like. So the elastin, people often talk about elastin fibers, but elastin is much more complicated than that. It can form sheet-like structures, struts. All of this is all embedded in there. So it's not simple. Now, to get a little bit more insight of what the cells live in, this is an individual collagen fibers. This is the cell, and you can see that one cell actually lives in it only touches just a few fibers, so the effects are probably fairly local. And again, here's this, I just thought it's kind of nice to see, this is what the elastin structure, so it's really more of an elastin web than really a fiber And you can see here, sometimes support these interesting sheet-like structures too. Now, the first thing we did then is say, okay, based on work we've done in the past, other people like the Yoga Nathan Lab and Merriman Lab have done related studies where we actually can take a portion of the mitral valve leaflet tissue, create a little strip, and we just cyclically loading it one-dimensionally. So this was kind of a first ex vivo study just to try to get some understanding of how the cyclic loading environment affects the cells. And as you load this little strip of tissue, these are maps of the collagen fiber alignment, and you can get from actually, and with the basic way to read this is that warmer colors mean a more aligned fiber. You can see as you pull that tissue, and of course, fibers become more aligned. If you look at the this on a graph, you can really see there's a huge change between 20 and 30% strain. So the physiological range is around 10%. So it's really kind of down here. Okay, now what we find when we do this is that this is after the control, and then which is what happens when we explant it from a sheep, and this is after 2 days later. Around 10% strain, nothing happens. So that's, and at 20%, a little bit of a drop, but not too much. So that suggests that the physiological range is probably somewhere in here. If you just don't deform the tissues at all, you actually get a drop in the soluble collagen production. Now, this is actually very important finding. In fact, it's actually even more important than this one here on the right, which I'll talk about in a second, because it says that in order for the cells to maintain their normal functional state or the normal phenotypic state, they have to be cyclically loaded. They need that deformation. If you unload them and just keep them in a living, tissue culture bath, they actually will their functional levels will reduce. We also, as I will show you, think that this is actually very important for understanding what happens to these cells post-repair, because as I'll show you, those the valves become less loaded. So we think actually part of the problem is they become less stimulated, therefore they reduce their ability to synthesize collagen and other extracellular matrix molecules. Now, the other thing you want to be very careful here is that at high strains, that is at hyperphysiological strain levels, that they become the opposite. They actually become hypersynthetic, and they really kick out a lot of collagen and lots of other things that are going on in there. And that actually is also very interesting. It's very important for things like mitral valve clips and other clinical applications. Okay, so and just some other metrics, soluble GAGs change as well. Both similar to the collagen. And if you summarize all this up for the different layers, you find actually that the different layers respond differently. Although the atrial and fibrosa respond similarly, the ventricularis just doesn't seem to do as much, not as sensitive. So this one is a made load-bearing layer, and we think this is actually very important, both going up. Okay. Genetically, we've actually looked at what happens down is a lot. This is obviously regulated by underlying genes. And what we find, of course, that at the more you stretch it, the more genes get upregulated. Basically, and especially the ones associated with TIMPs, basically in both the promoters and inhibitors of metallomatrix proteases, as well as collagen and other markers. So the short of it is that there's actually a direct genetic correlation between what's going on, how much you stretch it versus how much what the cells get stimulated. So to kind of pull this all together so you can kind of understand, this is kind of this little diagram we put together where the cells like to be in this green zone. I don't know if you can see that, but they like to be kept at a aspect ratio about 3.5 as they're cyclically loaded. If you go above that, they become hypersynthetic, and if you go below that, they become hypophysiologic. Things get downregulated. So basically, this is the sweet spot where the cells like to stay in. So the next thing we did is say, okay, this is great, but we need to make something closer to what's going on physiologically in the heart. And we did this by developing this bioreactor that tried to simulate what we found. And we guided that system by the sonocrystal studies we did a few years ago, where we measured not only how much the valve leaflet deforms in vivo, but more important, or just as importantly, what happens when you excise it, because we found that the valve actually is pre-strained. That is, when the valve is open, it's not unloaded, it's actually pre-strained. And you can see that here, if this black box represents this position of the sonocrystals, this black is the excised state, the red is the in vivo state, and the blue is what happens when it's fully loaded. So the deformation that goes in the heart is actually between the red and the blue states, but ex vivo, it actually contracts down to the black state, and this is really due to the elastin in the valve, and it shows that the valve is actually pre-strained, and we incorporated this into our studies. And again, here's a nice picture of the biaxial system, and it, nothing too elaborate. The main thing is it can be mounted in a typical incubator. And what we found is that just like those uniaxial studies, we actually emulated the effects of the normal stress, but the main thing, the ring stress. So if you put a ring on it and basically imitate the deformations of the leaflet tissue with that, guess what? The soluble collagen rate goes down after 48 hours, exactly as we predicted from the uniaxial studies. And this actually shows that some initial findings suggest that ring annuloplasty, which is one of the major therapies, may be responsible for poor long-term outcomes because it tends to reduce that aspect ratio. And in fact, here's the ring, here's the normal, and here's the data from that uniaxial study. And I think the trends are actually very, very similar. So there's actually some support of a direct connection between what goes on in surgery and what's going on biologically. And these are just some other metrics we looked at, alpha SMA expression and so on. And of course, the alpha SMA being one of the major markers of these cell types drops profoundly. And actually with the normal stress actually is pretty close to the post-mortem control, but we can see that going way down. Okay, pulling this all together, what does this find? Well, it says that if you put a ring on, you decrease the deformations of the tissue. Based on our in vivo, our computational model, this reduces the aspect ratio of the cell during the cardiac cycle. This is and then deep down in the cell level, you go from this physiological down to this hypophysiological state, excuse me, where things get down relegated. And this may happen over many months, that the tissue becomes degraded, and then undergo other changes that help lead to the recurrence of regurgitation after surgical repair, even if the patient responded very well to the treatment right after surgery. And this is just a little bit more information on how we linked it all, a summary of how we linked it all together, many levels of measurement, many integrated data. Okay, now, this is all nice and wonderful. And again, here's our super duper multi-scale modeling map. So this is a lot of information, a lot of interesting things, but again, how do we actually translate this to the clinic? And we need to do that. We need to go from this level down. We need to integrate it up to the organ level a little bit more closely. And we did this first by going back to one of the things that you can do these days is to look at mitral valve geometry right from clinical images. These are 3D ultrasound images taken from transesophageal. That's where the patient will swallow a probe, and they keep it, doesn't go down to the stomach, it stays in the esophagus, and then they can image the heart and the valve. And this is how we're actually able to get the surface reconstructions. We can then mesh it and create these beautiful in vivo maps of the mitral valve leaflet, very high fidelity maps directly from patient-specific information. Okay, now what we did then is that we looked at actual human clinical data. This is from something called the Cardiothoracic Surgical Trials Network, and this is the way it worked. We actually got the 3D echo in the open and closed state before surgery. Then they underwent ring surgery. Then we saw what happened immediately after surgery. And then we looked at 6 months after repair. And we had two groups that showed, we basically said one group that said they were successfully repaired, or the other one that mitral valve regurgitation reoccurred. And then we actually took that information, created the clinical image, and then actually came up with this cool technique to estimate the deformations of the leaflet tissue directly from the in vivo data without any physical marker. So we can use the clinical data directly. And this is a very rather involved technique. And the way we do this is that we started out with the open configuration and developed a finite element based morphing technique. It's not a finite element model directly, but it's a morphing technique that allows us to take the open configuration and push it into the closed configuration and gradually applying all the different things that you need to do, the annular displacement, pressurization, and shape enforcement. And if those of you are interested, I'll send you the paper. It's the technique is too lengthy to describe here. But basically what we do is we use a Fourier field to push those leaflets into shape, and it actually produces an extremely accurate result. What do we find is that this is the non-recurrent valves, this is the recurrent valves. Now, one of the first things you notice is that the non-recurrent valves are much smaller than the recurrent valve. And when we look at the radial, this is the deformations, and you can see here that they deform very differently. And what we then look at is we then looked at these different regions. Now, these are so-called the Carpentier segments. This is simply the anterior leaflet here, the posterior leaflet here, and then we split them up in these 3 segments. So we kind of peel the valve open so you can see what's going on. And what you find is that in the recurrent valve and the non-recurrent valve, initially they look similar, but if you look more carefully, you'll find actually there's some very interesting deformation patterns. And if you look a little, and particularly in these commissure regions, these are, and where we can see here that the recurrent ones deform very differently in these regions than in those in the non-recurrent valves. And if you pull this all together, you find that in these different segments, there actually are some interesting trends, mainly in circumferential direction, that we find that the recurrent ones deform quite a bit less. And this is, again, right in line with our hypothesis that that part of the reason why you get recurrence is these underlying biological changes because of the change in the way the leaflet deforms. And I think this is just a nice way to visualize what we found. This is what the leaflets look like, again, in that peeled open configuration in the non-recurrent and then in recurrent. And you can see here is that the recurrent are normally, they're larger, but they mainly seem to be stretched out more. Okay, so we lined up the annulus, and there's some changes up here, but it's really all of this down here. Okay, and they seem to be really stretched out quite a bit more. So again, when we modeled this with our technique, we did the same thing that I showed you before, but now what we did is we looked at the open state in the pre-MI after 8 weeks, and then we actually were able to morph all this together. And what we found was really interesting. In the pre-MI state, we found that after 8 weeks, the valve really stretches out. And in the radial direction, it also gets stretched out. So the valve seems to be able to deform plastically. That is, it's a permanent deformation. And then when we see this with respect to the post-MI state, we find that the overall deformation patterns do change, but especially in the circumferential direction. Looking at the tissue properties of we did this in sheep, by the way, and we explant the tissue. Again, we find that it's just like we saw in those earlier studies I showed you, that there's just profound changes in physical properties. So when we look at this biomechanically, we want to say, what are some of the tissue level responses? And then we find, and again, we talked about before about there's a very specific structures and architectures and mechanical properties of different layers. And we're looking down at what goes on in the valve, in looking at individual collagen structures and trying to get a handle what's going on there. And then we come up with this mathematical model for that. And the basic idea is to look at how the individual fibers of the different layers all work together to create these tissue responses. Now, the main thing you guys want to recall is that collagen, which is the main structural protein in the valve and other tissues, is undulated in its relaxed state. So when you pull it, it has a linear response, but there's this slack that it gets in. And this is what we think is what's changing. And that nonlinear stress strain curve that you've learned in class is really due to this recruitment of the collagen fibers and not to the properties of collagen itself. And when we get back to this, go back to that data and applied this model to that plastically deforming tissue after 8 weeks post-MI, we found that the collagen modulus didn't change very much, but all of this was explained by the changes in the tissue structure. And I think even if you don't get all that, this is basically what we think is happening at the tissue level just after myocardial infarction, let alone what happens after ring annuloplasty, is that pre-MI, the collagen is actually nicely undulated and deforms nicely. After myocardial infarction, the tissue will plastically deform, and that fiber actually straightens out. And that explains completely what we found both in vivo and in vitro. Okay, so to summarize, this is what we think happens in the heart after MI to the valve, is that there's a pre-MI state, everything's working great. there's an infarction, and then this is out to several weeks later, the leaflet actually will stretch out because of the dilation of this ventricle. And that dilation is permanent. It's non-recoverable. That's why I like to use the term plastic. And then you can see this diagrammatically here. If this is what the leaflet looks like pre-MI, post-MI, it stretches out by amount in this dark brown region. Okay, again, looking at the using our in vivo method, we actually want to be able to try to predict this. Now, one of the things that we want to be able to do is to predict what happens this in a actual model. And one of the things that's actually difficult to do in modeling the mitral valve is that, this is actually a beautiful movie from the Visible Heart Lab up in the University of Minnesota. And here's the aortic valve, here's the mitral valve. And again, you can see how taut those leaflets are. That's that pre-strain. But the biggest problem are these cords. If you want to model the mitral valve, how do you model the cords? Well, if you look at in vivo, you can get the leaflets pretty well, but you can't get the cords at all. Okay. And that's a big problem because these cords there's a very major part of the leaflet structure, and they're not put in any kind of random way. So we developed this method to look at, basically we used in vitro micro CT reconstructed ovine valves. And this is a very elaborate study, but basically what we're able to do is to determine a simplified approximated structure where we combine the origins. So we went through the ground truth, we then did constant cross-sectional area, combined no branches, and eventually combined. This is like the simplest model. And it's actually worked pretty well. And we came up with an optimal density where the error is pretty close, okay? And here's an example of what the actual mitral valves look like, and here's our approximated methods. We do know what the shape of the annuli are, because those are reconstructed from imaging. And then we actually put all this together in a computational model of the mitral valve and was able to take the pre-repair information and predict the post-repair very accurately. And you can see here, for example, the radial strains are much larger in the post-repair state, and this agreed very closely with our results. And here's just an example of how close this model worked. And these are some of the error maps, which generally the error is actually really quite low. So this gives us a lot of confidence that we can do this to take patient-specific information and come up with computational models to predict what's going on. Now, again, one of the major challenges in prediction of post-repair success is be able to do what happens to the leaflet after the repair. And going back over this plasticity model, we did this initially here and here. And the idea is that can we bring this model in to these clinically relevant data and models? And we did this by modeling this plasticity. This is very involved. We developed similar models for the soft tissues for prosthetic valves. But the basic idea works is that we actually can predict how the connective tissue fibers convect and change their responses as the tissue deforms. And when I say deforms, it means how it plastically deforms. And this is a very elaborate, I don't have time to get into this today, but it's all been published and it's really quite a nice model. And this is what we're able to do. We're able to take those reconstructions. This is the pre-MI. This is what happens if I just apply the changes in boundary conditions of that pre-MI geometry. And this is what happens when I incorporate plasticity. And that plasticity actually makes a big difference. So it's actually very necessary. And then we're able to take this and say, this is the normal valve, this is the IMR state, and then we can predict what happens to the valve in IMR and then also after valve repair. And this just also underneath this is what happens to the connective tissue structure. This is what's called total fiber recruitment. And we're actually able to show how much this color scale gives you the amount the fibers have been recruited. And what this means is that in the normal valve, in the open state, because of the pre-strain, there's some recruitment, but most of the tissue is in blue state, which means that there's a lot of structural reserve. But after it's closed, the ischemic mitral valve tissue becomes much more recruited, much straightened out, and that structural reserve is gone. So that means the stiffness of the tissue goes up, and again, that's going to change how it responds. Okay, so just to kind of wrap up, where are we going next? Well, again, this is ischemic mitral valve disease is really not a mitral valve disease. It's a left ventricle disease. And what the other part, what causes this is continued changes in remodeling the left ventricle itself. So the way we did this is that we went back and we developed this very detailed model of the ovine heart, and we did this with the Visible Heart Lab. And we actually took a single ovine heart, got its fiber structure with diffuse intensity imaging, got volume and pressure measurements, and even if you can see this here, we got electrophysiological measurements as well. And this allowed us to create a very comprehensive model from a single heart. We then were able to put a mitral valve inside this heart, and you can see here using the techniques I showed you earlier, and actually reconstruct it and also then predict this area, which is called the regurgitant orifice area, which is how much the valve can leak. And if this, what's supposed to happen, of course, is that this is closed. And the idea is now we can connect what's happening to the ventricle to what's going on in the valve. And we looked at different infarcts from both these different locations of the infarct. The most common one is this so-called posterior basolateral, and we also looked at the fiber stretch as we go along. And what we're able to determine is that which of these arrangements produces the greatest amount of regurgitation as initial test. And we found, of course, that this actually works quite nice, and we're able to predict that pretty well. We also found that if we put pre-strain on the valve, that if you do, and this is what the in vivo strain measurements are, you get the best agreement. If you don't put in this pre-strain, it doesn't agree very well. And if you put different levels of pre-strain, it works a little better. But if you put the physiological pre-strain on, you get a very close agreement. So that pre-strain is actually very important and will change with the valve. Now, to just kind of summarize all that up, if this is the regurgitant orifice area, that's how much it's leaking, if you will, with respect to all these different infarct types. We actually were able to show that the infarct, this posterior basolateral location, produces far and above the largest orifice area, and actually clinically is observed as the grade. The grade is the level of IMR, and this grade shows you how far it's going. So we actually get a direct connection to what's going on clinically. So just to kind of pull it all together, we've done the kind of a whole multi-scale approach, looking at organ level, at the whole heart, down to the individual valve level, down to the cell level, and down to even to isolated cell studies that we've done over the years to try to really understand this whole system in a multi-scale approach. And you really need to understand things this way because these are really complicated systems. They're amazing how they work, but the good news is that it is understandable. So, and it is modelable. And I like doing computational models because they're the best ways to really test how well you really understand what you're doing. So where we're going next? Well, all of this is great science and really interesting, cool stuff to do. But clinically, we're moving toward using this to optimize repair. So there's repair, which is still growing popularity, particularly with these transcutaneous devices, but they still suffer from long-term durability problems. A very common therapy that's kind of gotten very popular in years is the so-called Mitral Clip, which you can see here. And these guys, the patients who get this so far, the 2-year data is really quite remarkable. It's done minimally invasively, and it's just a clip. But again, nobody knows what the long-term success of this is. And you can see here with the clip, it's obviously can produce focal points of stress, and they don't even know where where the best way to do this is. So that's kind of where we're going with that. So the whole idea is to be able to use our fundamental understanding as well as our applications-based models to predict best ways to do this patient-specific manner. So I just want to wrap up just by thanking all the funding agencies for the generous support, wonderful collaborators at the Gorman Cardiovascular Research Group, and also Giovanni Ferrari's group at Columbia, and of course, all the wonderful people who do all the work, who's been working with me here at UT Austin for the last 10 years. And it's been, we're actually looking forward now to doing both greater fundamental studies, but the computational imaging techniques now allows us to translate that to the in vivo environment. So I just hope you all enjoyed that little journey, and I'm certainly welcome to open for questions. So thank you very much.
Thank you very much, Michael, for that excellent talk. And so let me look at some of the questions that have been asked.
Yeah.
And so we have a question here from Sophia,
Yeah.
and she asks about the idea of minimally invasive methods for mitral valve repair or replacement, and if there are some challenges that are specific to the mitral valve as maybe compared to the aortic valve in terms of minimally invasive treatments.
Oh, absolutely. So the mitral valve, the aortic valve, in terms of transcutaneous aortic valve replacement, is relatively successful because although because you have most of the patients have a heavily calcified aortic root, and they can anchor the valve relatively stable in a very stable manner. In the mitral valve, you don't have anything like this, and you're dealing with anchoring it in the left ventricle as well as getting it in there. Which is often done either apically or through the atrial access. Sometimes they actually go through the jugular vein and go through the the atrial wall. But the biggest problem with valve replacement, excuse me, is not just getting through these technical challenges of anchoring and stent design, but the fundamental problem is the leaflet material itself, and durability of these tissues are still very limited. And remember, for a transcutaneous valve, they have to make the tissue substantially thinner so they can get it into a catheter. And that puts much even greater demands on these tissues. And so surgeons ultimately, depending on who you talk to, ultimately the ideal situation would be a durable repair, because then you don't have that issue. Minimally invasive techniques like the Mitral Clip and the Pascal devices and many other devices are really ideal because they can be done minimally invasively. They're generally very stable, but we don't understand how they are to be used optimally. So I think most surgeons will have an armamentarium of both replacement devices and surgical repair, but I would say less invasive or minimally invasive surgical repair is the way to go, particularly for the mitral valve, because it's so much more complex.
Great. So another question comes from Daniel Chaparro from actually Josh's lab, who's a doctoral student in Josh's lab. So he asks, I guess, a twofold question. First part is, how drastic are the changes or alterations to the ventricular and atrial pressures after mitral valve to the mitral valve environment post MI? So maybe we can answer that question first. How drastic are the changes to the pressures, the ventricular and atrial pressures post MI at the mitral valve location?
Yeah, so the ventricular pressures aren't really affected by this. What is affected is the flow of the atria. And so one of the big problems with mitral valve regurgitation is, of course, you get backflow into the left atrium when it's supposed to be filling, so it interferes with the filling of the atrium. So the short answer is it raises this pressure of the atria. But also the most important problem is that it causes backflow of blood back into the lung, causing congestion and increased fluid in the lungs, and ultimately increasing the load on the left ventricle, in addition to stroke. So it's a combination of many different factors going on. But to answer your question, it mainly affects what goes on in the left atria, and its flow and pressure, and it becomes very aphysiologic. The left ventricle, not so much. But what it also does, of course, is increase the workload on the left ventricle, which is already in a bad state because of the presence of myocardial infarction.
Great. And I guess another follow-up question from Daniel is the mitral valve tissue remodeling that occurs post-MI, is it solely attributed to the mechanical changes, or could there be other factors, biochemical or biological, that attribute to that?
That's a great question, and I think we're still learning about it. Let me say this. So what we know for sure is the following. In sheep models and ovine models, which are the best we have, okay, that we find up to an 8-week time point, that's as long as we've been able to take it out, that all the changes at the tissue level, in other words, the geometry of the its tissue properties, can be completely explained by the changes in the shape of the valve in structure. So it's actually like the fibers, everything's sliding, fibers are rotating, all that is happening in the valve. And our mathematical computational models basically show everything can be explained by that mechanism. However, we also have found very profound changes to what's going on in the cells. And what I think the question is, how long are we talking about? So for the short term, say 2 months. 2 months is a long time, but it's not a long time for tissue remodeling. You know, tissue remodeling can happen over many months or years. There's actually evidence from Bob Levine's group is that in patients who've had ischemic mitral regurgitation for several years, that there's inflammation and swelling and other continued pathological changes to the leaflet that are probably driven by the initial things that we've seen. So the short answer is it's more of a question of how long after the MI are we talking about? So far, everything we can do. One thing I'll tell you for sure is that when they do ischemic mitral regurgitation treatment, that valve is no longer clearly in the normal state. And that's another big issue as well.
Okay, great. And so I guess one more interesting question. This one comes from Dr. PM Raj, who's a faculty in our department. So he asks the question about the distension of the valve. Is there other conditions besides MI that could potentially cause alterations in the mechanical properties of the valve? And can there be any insights on potentially a wearable sensor that could maybe detect changes, like he mentions the seismocardiogram, so that provides sort of a daily monitoring or feedback to the physician?
Yeah, that's a wonderful question. So the short answer is there are ischemic mitral regurgitation is just one form. It's actually not the most common, but it's the most deadly. There are other ones called people with congenital defects. Used to be rheumatic fever was a big source of valve problem. In fact, my wife's grandfather had rheumatic fever when he was a child, and I think ended up dying from mitral valve at that time. This is back about 1980, at the time when the surgeries were not as well developed, and they said you had a 50-50 chance of surviving the disease or surviving the surgery, and he decided to try to survive the disease, but he didn't. Much of that, and then of course there's another big one, which is the so-called Mixomonous heart valve disease, which is what's so-called organic valve disease, where the valve leaflet tissue itself has become degenerative and prolapse happens. Those tend to be treated fairly well by current techniques, so there's this need for continued improvement. Now, I think the better answer to that question is how do you monitor what's going on? And this is a great one, because nobody's going to go in and want to get a transesophageal probe stuck down their gullet to get them imaged. They'll have transthoracic imaging, but a technique that they could use to monitor these guys in some type of wearable sensor would even be better, or something that you could just apply at home. That would be a wonderful thing to do, not just for a whole range of other valve diseases.
Great. So one last question, Michael. So this comes from Valentina, also from Josh's lab. So she wants to know, in regards to MI, and you've shown clearly how the mitral valve is adversely affected. What about MI in the context of the aortic valve? And are there adverse events that happen to the aortic valve? And what's your thoughts on why the mitral valve versus the aortic valve, and why would one valve get more affected as opposed to the other in terms of its changes in the mechanical properties?
Yeah, that's another great question. These are very good questions, by the way. You're smart people. So the diseases of these valves are very different. Okay, now, generally speaking, aortic valves generally suffer from stenosis and insufficiency. They can become inflamed and calcify. And of course, there's the bicuspid aortic valve disease, which, by the way, is the most common cardiac congenital defect, which these patients have probably a much, much higher chance of contracting that. The mitral valve, generally speaking, it's not that it never calcifies, it can, but it's generally not the issue. It's usually due to a degenerative disease, the Mixomonous valve, which is the source of that is still not that well understood, or like I said, it's due to disease of the left ventricle because of its tight coupling with it. Okay, so but in terms of, we have shown in a recent publication that the properties of the bicuspid aortic valve are profoundly different than those of the normal valve. No big surprise there. So the short answer is they're all made from the same fundamental building blocks, and they're built in their internal structures are actually quite similar, but there are very interesting differences between them because they do have very different functions and anatomies. So it's not surprising that their pathological processes are going to be different.
Very good, thank you. And so I guess with that, that concludes our seminar for today. I'd like to thank once again Professor Michael Sachs for taking the time to provide these insights, specifically on mitral valve alterations following MI. Wonderful talk. We learned a lot. Thank you, Michael. And that concludes today's seminar. Thank you very much.
Thank you, guys.